Hi, this is Mr. McLean, and I'm back again to take you through some of the material of section 6.6 of the Campbell biology. And we're going to be taking a look at how the cytoskeleton is set up and the different parts of that overall network of structures. So the three big types of protein structures that we're going to see in this particular area are listed down here for you. So we've got our microtubules, our microfilaments, and our intermediate filaments. So if we're going to rank those in terms of their size, the microtubules are going to be the largest, the microfilaments are going to be the smallest, and the intermediate would be kind of the intermediate ones, which would be in the middle. So what I want to do is I want to take a look at some of the main features of each of these and kind of show you how they help carry out some of the major cellular functions. Keeping in mind the combination of these three structures are going to work to accomplish a couple things, and one of those features is going to help organize our cell. So we're going to see how different types of cellular structure and activities are ultimately controlled by the formation of the cytoskeleton, and we're also going to see that those organelles are not just suspended in the cytoplasm, but rather that they're actually anchored. Okay, so as we kind of move into the overall jobs and the roles, we can see that there's three big things listed here: support, motility, and overall regulation. And we're going to kind of see how each of those different structures are going to allow us to work together to support these different objectives. So the first one that we look at is how we move things within a particular cell. And we mentioned before the concept of almost like a monorail type of setup. And in this particular image, obviously we have the real image down here in the bottom, and if we look at it, we can definitely see this not getting a good zoom in here. Okay, you can definitely see how this right here is going to serve almost as like a tube right through here, and along that tube, kind of working with it, this would be our vesicle. So it's a lot of times easier to look at the artistic sketch to get a better idea of what's actually happening. But our vesicle is, as you may recall from earlier sections, basically some membrane, which is going to have not only the phospholipids, but it's also going to have some of those surface proteins. And it's going to package up different types of proteins, and that would also connect to this structure here, which is a motor protein. And we're going to see some motor protein used later on. And then we invest a little bit of energy in the form of ATP, we're actually going to be able to mobilize movement along a microtubule. So this supports the idea that our cells don't just have vesicles randomly moving throughout. They actually have a set pattern as far as where they can go, and it has a structure that will support how they go from point A to point B. So kind of moving us into those three different areas, we talked about the diameter or the thickness of each of those different filaments. I like to look at this particular diagram right here. This is on page 113, and if we zoom in on it, I think it does a good job of highlighting the major characteristics of each of those three areas. So I'm going to kind of revisit that. So the first one that we have here is going to focus on the microtubules. And as we said before, that's going to give us our largest overall diameter, because you have, as you can see in this picture down here, a collection of these different tubulin proteins. And when we zoom in, we can see that the arrangement of them is going to be kind of a helical structure, as we can see right here when we're going through with that. So as they kind of coil up, and you have alternating types, because you have an alpha type and you have a beta type, it forms this overall tubular structure. And that tubular structure serves a lot of critical functions. So remember, the protein type is called tubulin, and the big things that we're going to see is maintenance of the cell shape. How this works to maintain a shape of a cell, if we look at it, and this is a really basic image of a cell, but you think about it, you have forces that exist on the surface of the cell, and they're trying to compress that cell. So one of the things that the microtubules do is they create almost like an internal network that helps resist compression. So in this sense here, we're not going to crush our cell under the weight of other cells and other structures. So it creates more of that rigid resistance to outside pressure. We've already talked a little bit about how they play a role in motility. I'm going to focus on that a little bit more. And when you think about the processes of mitosis or meiosis, and if we had something that was set up, so our typical image here where we develop our spindle apparatus, and if we're looking at something that's maybe into an anaphase of some sort, and we have our chromosomes hooked on here and hooked on here, what we're going to see is that these fibers right here that we generically refer to as the spindle apparatus are the microtubules. So that helps with that, and it's obviously going to help with the movement of different organelles. Now, if we compare that particular structure with the structure of what the microfilaments would be, and this is going to be a much, much smaller diameter. So if you're comparing, this with this, the diameter of this type of setup is going to be dramatically smaller. I always use the analogy of a filament inside of a light bulb, how thin it is. These are filaments of actin fibers. So you're going to have a double twist of those. And like I said, the protein type is known as actin. And if we look at the jobs of it, one of the things that we're going to find is it's going to also play a role in shape. Now, one of the things that this will do, if we look once again at a really basic cell picture, we have forces that are pulling the cell. Kind of that fluid membrane of the plasma membrane can kind of be stretched and pulled. This network of actin fibers that exist inside of here will help resist that pulling. So they're going to kind of bear tension, whereas the other one was going to fight compression. This is going to fight being pulled apart. We're also going to see some of the role of muscle contraction. So our muscle is going to definitely use actin and myosin. We're going to see the mobility of a cell. If you're looking at a plant cell, they have something called cytoplasmic streaming. And we also see them playing a role in cellular division. Our third category will be our intermediate filaments. And in terms of size, they're in the middle. And probably the biggest thing to remember about this group is that they are more permanent than the other two. So in other words, if you look at actin, and especially if you look at tubulin, we can assemble and disassemble that portion of the cytoskeleton very quickly. When you look at the type of filaments that are built in the intermediate section, they're going to be more permanent. They're going to even last even after the cell has died. So they play a big role in shape once again. They also form that basket that kind of holds the structure of the nucleus known as the nuclear lamina. So that plays a major role in the overall structure of the cell. So just to highlight some of those things that we said, microtubules shape the cell, they help move things, and they help in cellular division because they are part of the overall spindle apparatus. Now, one of the things that sometimes becomes confusing is when we start introducing the concept of centrioles and centrosomes, which we know are structures that are going to be found inside of our animal cells. So if we look at this picture right here, what we're noticing is our centrosome, which is kind of shown with this yellow cloud of sorts, is a region that is normally found very close to the nucleus under times when there's no cellular division going on. So if I'm going to pull up our picture of our animal cell here real quickly and kind of isolate and show you where that's at. But if we go right over here, let me see if I can slide this over, make this a little bit bigger. Okay. So right here, this is that centrosome, and it's right now somewhat close in proximity where that nucleus is at. Inside the centrosome, we have two of these things right here and right here. Those are centrioles. Okay, so centrosome is this whole region, but inside the centrosome we have centrioles, and you can see coming out of the centrioles we have microtubules. So those microtubules in this picture are anchoring and serving as a support structure. When we think about the process of mitosis, what we're going to see is that those centrosomes are going to break out, and you're going to have your centrioles. They're going to migrate to opposite ends of the cell, and once they get to the opposite ends of the cell, they're going to sort of serve as our organization center for our microtubules. They're going to come through there. So we definitely see it in mitosis and meiosis. We're also going to see microtubules in the structures of cilia and flagella, and if we look at this picture right here, we talked about the fact that let's look at the cilia, for example. There has to be a connection point. The connection point where it hooks onto the plasma membrane is called our basal body. So that's our anchorage point for either the cilia or the flagella. And what we want to try to do is we want to try to create movement by being able to kind of bend and allow for either a snapping or a whip action type of movement. Now, if you look at the closer structure as to what is going on in a cilium and then the basal body as well, if you look right here, our cilia are arranged in what's called doublets. So this is a doublet right here. The fact that there are two kind of got a line there. These are two microtubules, and they make up a doublet. And if you look at the sets, you got 1, 2, 3, 4, 5, 6, 7, 8, 9. So a lot of times you might see like a 9 to 2 kind of configuration, or 9 doublets, and then you have these 2 in the middle. Okay, so if you see anything that says like a 9 + 2 configuration, that's referring to the 9 doublets and then the 2 that are inside of here that make up the overall structure of our cilia. And if you look at this picture right here, this is showing us what the basal body looks like. And if you compare the basal body picture with the picture of what we saw with our microtubules, you're going to see that they're going to have a very, very similar alignment. In fact, the centrioles in many ways are going to be the basal body when they anchor into that portion. Now, we talk a little bit in class about how we can actually get that whip action taking place, and we refer to that as a dynon walking. So if you look right here, you have your anchorage, and the idea is that these motor proteins kind of walk past each other with the investment of ATP. But because they're anchored and they're cross-anchored, that's what that blue protein is showing. It's this cross-linking protein. They don't slide or walk past each other like this top picture, but rather they create a bending and a flexing, and when we release it, it snaps over and it lets us move. Okay? Just highlighting a couple of the other structures real quickly in our last few minutes of this video. We talked about, once again, the actin a lot of times being bundled into things like our microvilli. So when you look at something that has projections, so here you find the lining of the intestinal tract is going to have a lot of these different types of projections, and they're going to be loaded with actin. This slide focuses in on 3 different actions where actin is very critical. Of course, muscle cells are going to be loaded with actin filaments. So that's this orange structure right here. So the thin actin is right here, and then we have the larger myosin that's going to interact with that. And what they're going to do is they're just going to kind of like grab on, and it's going to pull past each other. So if you look at this image, it's showing you that you've got a section of muscle moving this way and this way. Excuse me. And as it does that, it's going to allow for the contraction. We also see, in some instances, we have what's referred to as an amoeboid movement. So here's our actin, and it's creating a pressure and a force, and it's pushing some of that cytoplasm. The other one is cytoplasmic streaming. So if you hear about cytoplasmic streaming, that's telling you you're going to be moving cytoplasmic material through this system.